Hello everyone, welcome to the channel Microbial World. In this video, we will be learning about Proteus bacteria. Proteus bacilli are normal intestinal commensals and opportunistic pathogens like coliforms. This means that Proteus species are found as a part of the normal human intestinal flora, but it becomes pathogenic when it leaves the intestinal tract and especially when it comes in contact with urea in the urinary tract. The name Proteus refers to pleomorphism. Pleomorphism is the ability of microorganisms to alter their shape or size in response to environmental conditions. It is named after the Greek god Proteus, who could assume any shape. Genus Proteus has 4 species: Proteus mirabilis, Proteus vulgaris, Proteus mixofaciens, and Proteus spenneri. Proteus is a member of Enterobacteriaceae. It is a gram-negative bacteria. Gram-negative bacteria has a thin peptidoglycan layer and it appears pink upon gram staining. Proteus is a rod-shaped short bacilli and it is motile with peritrichous flagella. This is a flagellar arrangement where the flagella is distributed all over the cell surface. Proteus is non-capsulated. This means they do not have a capsule, which is an external layer surrounding the cell wall. They are non-sporing, so they do not produce bacterial endospores. Proteus also produces a characteristic fishy odor in the urine of infected individuals. This is because this bacteria has the ability to produce the enzyme urease, which converts urea into ammonia. They are facultative anaerobes, so they can grow in the presence or absence of oxygen. Habitat. Proteus is naturally found in soil, water, decaying organic matter, and as part of the normal human intestinal flora. Species of medical importance. Proteus mirabilis. Proteus mirabilis causes 90% of all Proteus infections in humans. It can cause urinary tract infections, septicemia, abdominal and wound infection. Proteus mirabilis is also a secondary invader of ulcer. This means Proteus can take advantage of the infected tissue in the ulcer and colonize it after initial damage has occurred. They can also infect burn wounds. Proteus mirabilis causes chronic discharging ear, that is, chronic suppurative otitis media. This is a persistent ear infection that can result in ear discharge through a perforated eardrum. Proteus vulgaris is an important nosocomial pathogen, so they can cause hospital-acquired infections, which means these pathogens can infect patients who are hospitalized for other medical reasons. Proteus vulgaris can result in wound infection, urinary tract infection, and less commonly respiratory tract infection. Virulence factors. Urease activity. The enzyme urease contributes to the formation of kidney stones by increasing the urine pH. Fimbriae are adhesive structures that helps the bacteria to attach to the epithelial cells of the urinary tract. Endotoxins and hemolysins produced by Proteus bacteria contributes to tissue damage and inflammation. Symptoms. Urethritis. Urethritis is the inflammation of urethra. The enzyme urease produced by Proteus bacteria hydrolyzes urea into ammonia and carbon dioxide, raising the pH of urine. The elevated pH leads to the precipitation of magnesium ammonium phosphate and calcium carbonate, resulting in the formation of struvite stones, a type of kidney stone. These stones can irritate the urinary tract, especially the urethra, leading to inflammation. Pyelonephritis is kidney infection. If Proteus spreads into the kidneys, it can cause symptoms such as fever, flank pain, and systemic symptoms of infection such as chills and malaise. Cystitis is bladder infection. It is characterized by symptoms such as dysuria, which is painful urination, and also frequent urination. Biochemical traits. Proteus is urease positive. It is non-lactose fermenting, so they do not ferment lactose. They produce hydrogen sulfide, particularly Proteus vulgaris. Indole test is positive for Proteus vulgaris and negative for Proteus mirabilis. Proteus is oxidase test negative, methyl red test positive, and Vogt-Prosker test negative. Citrate test is positive for Proteus mirabilis and negative for Proteus vulgaris. They can ferment glucose with the production of gas. They also have the ability to reduce nitrate to nitrite. Swarming motility. Swarming is a coordinated movement of large population of bacterial cells, allowing them to rapidly spread over a surface. On agar plates, Proteus exhibits a characteristic swarming growth, producing concentric circles due to cyclic waves of movement. Lab diagnosis. Culture on standard media such as blood agar or McConkey agar. Proteus species produces a swarming growth on blood agar. As they do not ferment lactose, they produce colorless, non-lactose fermenting colonies on McConkey agar. Biochemical tests such as urease test, hydrogen sulfide test and motility test is commonly used. Antibiotic sensitivity test can also be done to test the susceptibility of the organism to various antibiotics. The commonly used methods are disk diffusion method and minimum inhibitory concentration test. Treatment. Commonly used antibiotics include fluoroquinolones, cephalosporins, aminoglycosides and trimethoprim sulfamethoxazole. Management of complications include removal of indwelling devices such as urinary catheters and addressing any structural abnormalities of the urinary tract. Thank you for watching. Do subscribe the channel for more informative videos.